Hello viewers, today we will discuss the antagonists and our today's discussion or learning objectives are by the end of the session you will be able to discuss the types of antagonists that what are the different types of the antagonist and then the mechanism of antagonist, how the different types of antagonists they produce the antagonistic type of effects, and then we will discuss the clinical importance of these antagonists. So the antagonists, they are the drugs that they decreases the action of the other drugs or the endogenous ligand, and the majority of the antagonists they act on the identical receptor binding site as the agonist. But not all the antagonists or not all of the mechanisms of the antagonism they involve the interactions of the drugs and the endogenous ligands at the binding domain of the single type of receptor. So there are many types of the antagonists, but three are main important, that is the pharmacological antagonist, and another is the chemical antagonist, and third one is the physiological antagonist that we will discuss one by one. So this is the pharmacological antagonism, and pharmacological antagonists they are divided into two types, that is the competitive antagonist and the non-competitive antagonist. So the competitive antagonist means there is the competition of the antagonist with the agonist for binding to the receptor site, while the non-competitive has no competition with the agonist with the binding site. So more generally, this is the receptor, that the outer domain of the receptor, transmembrane domain, and the internal domain of the receptor, and the antagonists they come and bind with the binding domain of the receptor. They just occupy the binding domain of the receptor because they doesn't shows any intrinsic activity, they don't have any efficacy, and they only have the affinity, that is they have the attraction towards the binding domain of the receptor. They occupies the binding domain of the receptor and they prevent the binding of agonist with this binding domain. So this antagonist who have no intrinsic activity and that therefore they produces no effect by themselves, and although they are able to bind avidly to the binding domain of the receptor because they possess the strong affinity towards the receptor binding site. So if both the agonist and antagonist bind to the same binding site of the receptor, they are called as the competitive antagonist. So this is the situation, that is the agonist. It occupies the binding domain of the receptor. It produces the effect. If you want to produce the antagonistic effect, so if you increases the concentration of the antagonist, like if you increase the concentration of the antagonist, then this antagonist will finally displaces this agonist from the binding domain and occupies the binding site. And if in another way, if you increase the concentration of agonist, so when the agonist concentration is increasing, so finally it will displace the antagonist and come and bind with the binding domain of the receptor. So that's why we call it as the competitive antagonist, like prazosin, that is the it is one of the example, is alpha-1 blocker, and it competes with the endogenous ligand norepinephrine at the alpha-1 receptor binding site, which decreases the vascular smooth muscle tone and reducing the blood pressure. And another is the non-competitive type of an antagonist, that it just come and bind with this binding domain of the receptor. If you increase the concentration of the agonist, it will not displaces this competitive antagonist from the binding side because it is a permanent binding. That is the covalent bond, that is might be the van der Waals bond that between the drug and receptor binding domain. So whatever the concentration of agonist is, it will not displace this antagonist from the binding side. It is more permanent. That's why we call it as the irreversible non-competitive type of the antagonist. Another type of an antagonist which doesn't come and occupies the binding domain of the receptor, it just come and bind with the other side than the binding domain of the receptor. So we call it as the allosteric antagonist, that it doesn't compete for the binding domain of the receptor. It doesn't occupies the binding domain of the receptor. It just occupies the site other than the binding domain. So we call it as the allosteric antagonist. And so it allows the agonist to come and bind with the receptor and produces the effect, but in the presence of the allosteric antagonist, the agonist will produce the less effects. And the another type of the antagonist is the chemical antagonism. And this chemical antagonism or chemical antagonist need not involve a receptor at all. Thus, one drug may antagonize the actions of the another drug by binding, chemically binding to and inactivating the other drug. For example, that we have the well-defined drug that is the heparin, which is an anticoagulant, which is a negatively charged drug. And if we administer the protamine, which is positively charged drug, as the physiological pH, it can be used clinically to counteract the effects of the heparin. So in this case, the one drug antagonizes the other simply by binding it and making it unavailable for the interactions with the proteins which are involved in the formation of coagulation. So the heparin will lost its coagulant effect, and it will not produces the anticoagulant effects. So we can say that this is the chemical antagonism. Another antagonist is the physiological antagonist. Now the physiological antagonism, this is the antagonism between the endogenous regulatory pathways, and the many physiologic functions, they are controlled by opposing the regulatory pathways. So the well-defined, in this case, in the physiological antagonism, there are two drugs. Both of the drugs, they are agonist. None of the drug is antagonist. Every drug come and bind with its separate own receptor, but they produce the opposite effect. They nullifying the effects of each other. That's why we call it as the physiological antagonist. For example, that is in the bronchial tree, or in this lungs, we have two type of well-defined receptor. One is the histamine receptor H1, and another is the beta 2 receptor. For the H1 receptor, the agonist is the histamine. When it comes and binds with this H1 receptor, it causes the bronchoconstriction. And at the other hand, we have the agonist for the beta 2 receptor, that is a norepinephrine. And when this norepinephrine come and bind with this beta 2 receptor, it causes the bronchorelaxation. So the bronchoconstriction, which is produced by this H1 receptor, is being nullified by the bronchorelaxation produced by this norepinephrine. by acting on beta 2 receptor. So we can say that histamine is the physiological antagonist of the norepinephrine. And the several catabolic actions of glucocorticoids lead to increased blood sugar, hyperglycemia. That is an effect that is physiologically opposed by the insulin. Although the glucocorticoids and insulin, they are act on a quite distinct receptor effector system. So the clinicians, they must sometimes administer and advise the insulin to oppose the hyperglycemic effect of glucocorticoids hormone. So in general, use of the physiological antagonist produces the effects that are least specific than or the effects of the receptor specific antagonist. For example, to treat the bradycardia caused by the increased release of the acetylcholine, the clinician could use the isoproterenol, that is a beta agonist, and it increases the heart rate and causes the tachycardia. But the use of this physiologic antagonist could be least rational and potentially more dangerous than would use of receptor specific antagonists such as atropine. So in some clinical condition, physiological antagonist is very much useful, but the pharmacological antagonists, they are much more important and more commonly used for the treatment of different diseases. Thank you very much.